My name's Susan Kabacia. I'm a research scientist at the National Museums of Kenya. And this evening I'll be presenting on mushrooms. And this is on the collection methods that are basically used by the citizen scientists. Two types of methods are used. And they're mainly manual collection and the digital collection. And on the manual collection, this is where the citizen collects data in the field or collects samples in the field and submits them to a research scientist, or submits the observations and the notes to the research institution. And the other method of collection uses the online data collection tools, where the observations and the notes are submitted online. And there are requirements for both the methods, and this is whereby the collector should have basic training on how to collect mushrooms in the field, and also to have the understanding of how the mushrooms look like. Secondly, we should be aware on the collection seasons, whereby there are months that are suitable for collecting data or materials, and this is mostly during the rainy months or seasons. The other thing is that he or she should have the knowledge on handling different types of mushrooms. This is because mushrooms are of different forms, and at the same time, it's also good because he or she can take a precaution, and especially for those mushrooms that are known to be poisonous or be toxic. The other aspect is having the knowledge on what information to document or what information to accompany the specimens, or rather the mushrooms that he or she has collected in the field. And it's very, very important to note that not all mushrooms are edible, and there's those mushrooms that are poisonous or toxic, and therefore, unless one is familiar with the edibility of the mushrooms, then it is important to take note so that he or she is not poisoned. The other aspect on digital collection, and it's a requirement, is having acquiring very good photographic images which are downloaded online using the data collection tools, where in this case or in this project we recommend the use of the iNaturalist. And this is where the photos are uploaded together with the notes on this platform, which is an online repository, where now scientists come and they can be able to identify the specimens, or further, or even for their own research. And this helps so that in case some species are not known within the country, or they are not people that are familiar, especially those who are collected with the specimen identity, then they can be identified by other research scientists around the globe, possibly at genus level, at species level, or even at family level. And before collecting samples in the field, there are also requirements. And one of the requirements is to acquire permits and permissions. And this is because mushrooms are found occurring in different entities, which are some are government protected areas, others are private farms, and it's important to have permission to access such places. When it comes to the collection, it's also important to have equipment and also to be well adorned for the field work. And this is because as far as the mushrooms are concerned, they grow in different habitats on different substrates. So there are tools that are important actually in obtaining the collections. Some require uprooting, some requires cutting off the substrate. It's also good to take images, and that is why we need good phones and a good phone with a good camera. It's also good also to have a camera if need be, and also, and that is not when need be. It's an important gadget, but as well, the phone can also play the part of a camera. And this we are going to see as we move on in our next slides. For the personal safety, it's important to have protective clothing. It's good to have gumboots. It's a rainy season, so it's important to have a raincoat. It's good to have a first aid kit in case of any form of an accident. Sometimes they are minor. It's important to have these gadgets in place. Then it's important for the collector to have a field guide. It helps, it plays a role in identifying with the different forms in nature, and also in very basic description or identification of mushrooms that are growing in the wild. And it's also important to have the knowledge on where the mushrooms are growing. They grow in different places. They grow in different locations. So we find some in the forested area, some in plantation forests, and also their feeding also is different. Like for the saprophytic fungi, they've been found growing on wood. There are some that grow in association with specific trees, or rather, there's some trees that cannot grow without association with mushrooms. So it's important when documenting information that this information is captured. Some will be growing on living trees, and this is because of their saprophytic, of their parasitic nature. And then there's a process now of collecting mushrooms, and it is very key for photos to be taken and good photographic images. And this is of the habitat where the collection is being done, because the site ranges the grassland, plantation forest, indigenous forest. This is key when identifying and also when associating some mushrooms in the habitat where they grow. And also on the specimen, it's good to get good images showing all the views of the specimen. It will be on the upper part of the cup, under the cup, the style. Every detail of that specimen should be captured in a photograph. Then also, it's also important to take the specimen with the scale where a ruler is placed, and it can show the dimensions of a given specimen. Then when it comes to the label, it's important to place the label close to the specimen, because this reveals a lot. It reveals where the specimen was collected, the number and the date, and this helps when placing a specimen with the right data. And consideration should be given when collecting samples, that they should be of good quality. This is specimens that have not decayed, the specimens that are not very mature. The other aspect is on the developmental stages. It's good when collecting the samples to collect all the specimens with their different developmental stages. And also, it's also important to understand that because of the different sizes or different nature of growth, how to collect. And for the very small mushrooms, they can be uprooted very well, making sure that all the details about that specimen have been captured. Then ones that are growing on wood, they can be sliced well so that a whole specimen is obtained. Then for the soil dwelling, they can be uprooted well using a scooper, making sure that the base, or rather the site, is not damaged. It is uprooted completely. And when it comes to the sample size, you find that because of the different sizes, the quantities also matters, whereby for the very small sized mushrooms, 15 to 20 specimens will be okay to collect. For the medium, 5 to 10. For the large ones, at least 2 specimens will be enough. Then this is now the information that should accompany the specimens. And this is normally in form of a sheet. And this sheet contains the collector's details, which includes the collector's name, the field number that the collector has assigned to his sample or her sample, the site details, is which area was this mushroom collected, what was the land use, is it indigenous plantation, is it a farmland, was it growing on soil, on litter, on stumps, and what were the associated plant species. Then the other aspect that should be considered is the specimen itself, the crate. What color is it? What texture? Does it have odors when smelled? Does it change? Does it have color changes when bruised, or rather when it is cut? It is important to capture such details. Then labeling is key, as we said, and this is because the label carries important information, and it should be tied to the specimen. And the label or the label and the specimen should be placed in a paper. We normally use the greaseproof papers, and then the specimen is placed in the basket, ready for transportation to the research institution. The collection should be submitted, if possible, the same day. And if it is not possible, then they should be dried or stored in the fridge. Drying should be at 40 to 45 degrees centigrade. And if kept in the fridge, it should not be more than 2 days. Before drying, it's important to take a spore print. This is whereby the cap of a mushroom is cut, is placed on a black, white, or green paper. It usually forms overnight. It usually forms overnight or after a few hours, and it is placed very well in a Ziploc paper and stored together with the other specimens. Then once the specimens have been submitted to a research institution, then further identification takes place. The specimens are further described, they are dried, they are stored in airtight containers, and after that, they are curated for future research. For the submission of observation online, then after the collections have been made, they have been submitted, then photos can be uploaded together with the notes using the recommended online data collection tools, and in this case is the iNaturalist. And the images should be of good quality, and the site and the specimen details should be conclusive. I want to acknowledge the National Geographic for funding this work. I want to acknowledge the National Museums of Kenya and the University of Nairobi for the research that has been carried out in this project, and also Kenvo for helping us, and for the team that you have worked with in this project. And finally, I want to say thank you to all for listening.